Two. Hello, everyone, and welcome to the Quantitative Biosciences Institute, or QBI, here at UCSF, the seminar series. First, I'd like to thank Nevin Krogan and Jacqueline Fabius for sponsoring this series, and to the QBI events team, Gina Nguyen and Alexa Rocourt and Carolina Lenzi for doing all this great behind the scenes work. So thank you all very much. I am pleased to introduce today's guest speaker, Gilles Gasser from the Paris Sciences Lettres University. Oh, sorry, excuse my French. It should be a much better accent. You're probably thinking it's 11 p.m. in France, right? But we're lucky enough to have Gilles here in San Francisco, so he's not having to talk at 11 o'clock at night. Let me just give you a little bit of background on Gilles. He was born and raised and educated in the French-speaking part of Switzerland. He received his PhD in supramolecular chemistry at the University of Neuchâtel in Switzerland. He then did a postdoc at Monash University in Australia in bioinorganic chemistry, and then he did another postdoc with Professor Niels Metzler-Nault at Ruhr-University Bochum, Germany, in bioorganometallic chemistry. In 2010, he became a Swiss National Science Foundation Ambition Fellow at the University of Zurich, where in 2011, he actually became assistant professor. And then in 2016, he moved to Chimie ParisTech at the Paris Sciences Lettres University to take the PSL Chair of Excellence. And this is where he currently manages a very active lab. Gilles is the recipient of several fellowships and awards, including the Alfred Werner Award from the Swiss Chemical Society, an ERC Consolidator Grant, the Dean Chemistry Journal Award, the Drucker Award for his contribution to cancer research. And then recently he received the European Bioinorganic Chemistry Medal. Wow, congratulations, Gilles. I'm very honored to be one of his collaborators through the QBI Curie PSL funded project. And today he's going to be talking about metal complexes and diagnostics and therapeutics. So Gilles, please.
Thanks a lot, Judy. I will start sharing my screen. Okay, I hope you can see my screen and my presentation and the pointer. Thanks a lot, Judy, and thanks everyone for making my stay today at UCSF really so nice. It's really a great pleasure to be back in California. And what I would like to do over the next 45 minutes is to show something maybe which is not known to everyone, but it is the use of metal complexes in medicinal chemistry. And I would like really first to acknowledge all the students in the group. This picture has been taken a few weeks ago in Paris. We had to add a few students who were not present during this day using Photoshop, and that's why you have also funny pictures here. What I would like, if I can, oops, I'm sorry, I cannot change slides for some reason. I will stop sharing and share it again. Sorry for the problem. It happens here. Okay. Yeah, what I want to show you here on this slide is six different metal complexes which are heavily used in medicinal chemistry. And maybe some of you have already received some of these compounds. Today, if you are diagnosed with cancer, you have more than 50% chance of getting a platinum-2 complex, okay? More than 50% chance to get a compound like cisplatin. Today, if you have to go for an MRI and you need a contrast agent, you will receive a gadolinium-based compound like this one. And what I was discussing during lunchtime, it is really interesting to see that you are getting about 10 to 20 grams of this compound during a scan. And this emphasizes really that such compounds are not toxic, okay, if you receive so much of a compound. Organophine is used to treat some condition like arthritis. If you have to go for nuclear imaging, you will get a technetium-99M. If you are diagnosed with a certain types of leukemia, you will receive actually an arsenic-based compound, trizenox. And this compound has really helped patients to survive this disease, because now, thanks to trizenox, 95% of the patients are surviving this disease. You have maybe probably received a mercury-based compound like thimerosal, which is used as a preservative for vaccine. And this is very interesting because quite often people are believing that metal complexes are only toxic. However, they can really play an important role in medicine. And the reason is metal complexes have really additional properties or different properties than purely organic compound. If you think about cisplatin, the mechanism of cisplatin is the patient is injecting intravenously the compound, and because of the high concentration of chloride in the blood, the compound will stay as is in the blood and able to enter into cancer cells. In the cancer cells, the concentration of chloride is lower. You will have an exchange between the chloride and water. So the platinum will become positively charged, the platinum complex will become positively charged and go to the nucleus, which is to the nucleus because DNA is negatively charged. And finally, it will bind covalently to DNA, leading to cell death. And this mode of action is really typical for metal complexes, is really unique for metal-based drugs. And this is why it is complementary to organic compound. You can also see that you have redox activity, which can play a role when you are using, for example, an iron-based drug candidate. You have two different redox states, iron 2 or iron 3. You can even have maybe others, but that's the two predominant one. Finally, something that I would like to highlight is this higher structural diversity. It was really shown by Eric Meggers using a very simple concept. If you take a carbon and you put 4 different substituents on the carbon, the best you can do in terms of diversity is 2 different enantiomers, like the 2 hands. Now, if you take a ruthenium atom and you put 6 different ligands around the ruthenium atom, you can get 30 stereoisomers, meaning that you have a bigger diversity, okay? And this is very nice if you want, for example, to selectively target enzyme. And this was used by Eric Meggers to target specifically kinases. You have also catalytic properties. For the moment, metal complexes are heavily used in organic chemistry to do catalytic process. However, this can also be done in cells. And finally, something we are also working on is the fact that many metals have radioisotope, which is very interesting in nuclear imaging, for example. Now, the group is always working with metal complexes. That's a different picture just taken before COVID. You have Pierre and Marie Curie in the back in their garden, just next to Chimie ParisTech. And the metal complexes that we are preparing by ourselves, we are using in different areas of medicine. And today I want to talk about phototherapy and fungal infection. So the first project linked to photodynamic therapy, I really would like to highlight today the work of Johannes here, Martha, and of friends. Photodynamic therapy is a medically approved technique to treat certain types, for example, of cancer or infection. It relies on the intravenous injection normally of a photosensitizer, and you wait until the photosensitizer is reaching the target you want, for example, the tumor. Once it is in the tumor, a doctor, a practitioner, can irradiate the tumor using light so that it excites the photosensitizer, which will reach a triplet state. And this triplet state can react with the oxygen present in tissue to form reactive oxidation species. And this will kill the tumors, the cells. Now I insist that the photosensitizer that you are injecting to the patient, per se, it is non-toxic. Okay, it is only when you shine light, or you, the medical doctor, shine light, that you will get a cellular toxicity. This is definitively of interest if you want to avoid, for example, certain side effects. And this techniques, this photodynamic therapy that we call PDT, is really not only an alternative to treatment, but I would say a complementary technique to chemotherapy or radiotherapy. Now, in a chemistry point of view, the different photosensitizers which are approved are generally based on porphyrin phthalocyanine. That's the chemical structure that you can see here. And you can really see here that a palladium-based compound has been really recently approved to treat prostate cancer, okay, using PDT. And this is very nice to see that even palladium that you believe is only used in organic chemistry is now also on the market as a drug. Now, if you see that all the compounds are coming from the same family of chemicals, you can also assume that they will have quite often the same drawbacks. And the drawbacks are normally a poor water solubility, synthesis, which is quite complicated, as I was discussing with Judy before, but sometimes the compound can take very long to be excreted, okay, from the patient, meaning that the patient has to protect himself or herself from the light to avoid, to get sunburn. And technically, the compounds that you are injected into the patient have a relatively low cancer cell selectivity. However, the selectivity is given by the light, because you are shining light only on the tumor, you have a type of selectivity. However, it will be nice to have a second level of selectivity with a compound which is mainly going only into the tumor. And this is why we have decided, like others, I have put only a few key players in the field to use metal complexes. And the one we were extremely interested in are ruthenium-2 complexes, polypyridyl complexes, because you have polypyridyl complexes all around the ruthenium-2 center. And the way we were selling this is that this type of compound produce singlet oxygen, that's the agent which is really killing the cells. They are not photobleaching, meaning when we shine light, the compound is not destroyed. It's very easy normally to make such compound. They are relatively highly soluble because they are positively charged. They are inert, relatively non-toxic, I insist on this point, and cheap. And cheap, it's because you don't need a lot of synthetic steps to make the compound. And the highlight is really TLD 1433 from my colleague Sherry McFarlane. She has brought this compound already to phase 2 clinical trial as a photosensitizer for PDT to treat bladder cancer. And this is very interesting to see that such a compound can be injected into the bladder of patients with certain types of cancer, and through irradiation with green light, they can really not only save the bladder, but really save, not only save the patient, but also save the bladder of the patient, and you avoid really a very painful treatment, like even the removal of the bladder. And this is very nice and really a huge encouragement to keep going in this field of research, and I really hope that this compound will be approved in the very, very near future. Now, in terms of photophysics, I won't too much insist on this, but if you look at this simple compound, you can really buy it from any companies. You see that this compound has a maximum of absorption at about 450 nanometers, which is fine to treat superficial, tumor. But if you would like to treat deep-seated tumor or large tumor, you know that in order to have a deep penetration into the skin, you will need to use light with higher wavelengths. And this is what we wanted to do in this project. We wanted to shift this spectrum towards the red. And in order to do this, we receive some money from the ERC, and I could hire Johannes, Franz, and Martha, who all passed the PhD just before the COVID time, and Martha during COVID time. That's why she had to do this by Zoom. And I really thank them, all of them, and I will present their work now. And we had two main ideas to do this. The first idea was based on computer first. It means that we wanted not to make 500,000 of compounds and see which one was the best, but we wanted to rationalize which compound would be the best, and then prepare this compound and see if they were active or not. And to do so, we decided to use two types of ruthenium complexes. We have decided to take two Phen ligand and to change here the R group. Okay, we have systematically changed the R group, did calculation so that we can guess what will be the UV spectra of all the compounds, so that we can see which one are shifting the absorbance of the compounds towards the red. And here we have done exactly the same. We knew already the results here, so that's why we have only chosen those two here. And we have taken here not a Phen, but a B-Phen group. We have done all the calculation with Ilaria Ciofini. I will discuss more about this afterwards. And then we have intentionally made the compound so that we can check if what we have found was fitting with the experiment. We have done all the photophysics with Philippe Gonner at Chimie ParisTech, and we could indeed see that some of the compound were starting to absorb not anymore at 450 the maximum, but really more towards the red, exactly how we wanted. We could also see that each of the compounds that we have made were producing singlet oxygen, and singlet oxygen is really the species which is killing the cells. You could see also that we have, quantum yields of about 57, 69%, and these values are really comparable with the compound which are currently on the market. So until now, it was really, really nice. We had exactly what we wanted, compounds which were absorbing more towards the red and which were producing singlet oxygen. Now, so it started now the biology. We can also doing this in our lab. And it looks a bit boring, but I want just to explain exactly what we have done. First of all, we have taken all the compounds and we have irradiated them at 480 nanometer to see which compound were toxic. And afterwards, we decided to take only one and go to see if they can be irradiated at higher wavelengths. Now, as I told you, you really want a compound which is in the dark, non-toxic. We have used different types of cells. So you have colon cancer, you have cervical cancer, you have glioblastoma cancer. This is a non-cancerous cell line. And what you can directly see is that the first 5 compounds here are non-toxic in the dark. But once we irradiate the compound, we did not see really an increase in the IC50 values. There was really no effect of light on the compound. This was really, really disappointing. However, for the last 2, also some were a bit toxic in the dark. However, the phototoxicity was really increased upon light irradiation. So this was very nice. However, there was a bit this frustration that we have calculated all those compounds that we know should be active, but they were not. And this is where it is very interesting to use metal complexes, is because we could rationalize this result with the deceiving results with inorganic chemical biology. Because we were giving to our cells a ruthenium-based compound, none of those cells have ruthenium, okay, normally. It's only if we give them our compounds that they will have ruthenium in the cells. And we could determine the amount of the ruthenium in the cells using ICP-MS. And you see that the 5 first compounds that we have tested are going really, really poorly into the cells, contrary to 6 and 7. And the reason why those compounds was phototoxic is because you had more ruthenium in the cell, so that when you irradiate the cell, you could produce singlet oxygen and kill the cell. And this was explained thanks to the presence of a metal. So technically, all the theoretical work that we have done were making sense. Okay, we could really have compounds which were absorbing towards the red. However, the calculation doesn't tell you if the compound will enter the cells or not. Now we have only chosen one of those compounds, the best of the series. It's this one, very simple compound to make. And you could see that now we could really irradiate even at 595 nanometers, so nearly 150 nanometers more than before, and we could still get a very nice photo index. And the photo index is a value without unit when you divide the toxicity in the dark by the toxicity upon light irradiation. We could really see that the compound that I have shown you just now was very photostable, that we can shine light. We have used exactly the same settings than for the cell experiment. In comparison, the protoporphyrin 9, which is an approved photosensitizer, you see that the spectrum is really changing as soon as you shine light. Okay, you have really a lot of photobleaching, contrary to the ruthenium complex. We could do a lot of confocal microscopy because the compound is phosphorescent. This is our compound. You can see that it is really accumulating into the cytoplasm. We have used different dyes to see if there was some colocalization, for example, in the nucleus or in the mitochondria, but it was not the case. The compound is really going into the cytoplasm. We could also use this compound, we could also test this compound on spheroids, those mini tumors of about 0.5 millimeter in diameter. And you could see that the compound was really killing those spheroids upon light irradiation at 595 nanometer. And I'm very pleased to say that we have also done some in vivo. We could not see any toxicity of our compound. And we are currently performing efficacy of the compound in vivo using mice now. So we hope to be able to say more about the efficacy in mice over the next few months. That was the first idea which has worked. We could really push the absorption of the compound towards the red, but we wanted even to go further, towards the near IR. Is it possible to use such compound towards the near IR? And to do this, we have decided the two-photon photodynamic therapy option. What is it? Normally, when you want to reach an excited state, you can use 1 photon, for example, at 400 nanometer. You reach the excited singlet state, and after intersystem crossing, you reach the triplet state, which is necessary to react with triplet oxygen. However, you can also use 2 high-energy photon at 800 nanometer, and if it happens exactly at the same place at the same time, you will be able to reach the same state, the excited singlet state. This is very nice because if you irradiate at 800 nanometer, as I have shown you before, you can go really much deeper into the skin. What has been shown in the past is that some ruthenium complexes have this ability to absorb 2 photons to reach this excited singlet state. This is Professor Göppertal-Meyer. She was a German-American scientist, and she gave a name to this unit, the Göppertal-Meyer, of the 2-photon cross-section. This is a value which tells you how good a compound can absorb 2 photons. And you can see that all the ruthenium complexes which have been reported so far had a 2-photon cross-section between 40 to 250 Göppertal-Meyer. And the idea is to have compound with a higher 2-photon cross-section as you can. In the past, a group in Lyon in France has also shown that, that ruthenium complexes, as well as other metals, if you increase the delocalization of the complex, you can start to have really much higher 2-photon cross-section, up to a few thousand Göppertal-Meyer. However, it has never been used in PDT. And what we have done, again, it was to team up, first of all, that's Johannes, that's him who has done all the work. He's now in San Diego, he's a postdoc at UC San Diego. And what was interesting about Johannes, he really wanted during his PhD going to China. So he went to Professor Hui Chao at Sun Yat-sen University in Guangzhou to do some of the work that I will show now. And I'm really thankful to both of them. So what we have done, and it was Johannes who has done it in collaboration with Ilaria Ciofini, who is really a specialist at Chimie ParisTech in TD-DFT. We have once again, before doing all the compound, to look at which compound will have the better two-photon cross-section. Okay, we have done this by TDDFT calculation, and then Johannes has prepared all those compounds. You can see that we have done compound which was symmetrical and non-symmetrical with a high delocalization or non-delocalization. And what we could really show is that some of our compounds started to have a two-photon cross-section up to 8,000 Geppertel-Meyer, and this is very, very nice in terms of two-photon PDT. You see afterwards that when you have done the compound, okay, that's when Johannes has made the compound, you see that the UV-Vis spectrum of the compound are really starting to shift towards the red, up to 700 nanometers. This was very, very, very nice, exactly what we wanted. And we have done afterwards, as usual, the biology. It looks like a terrible slide, and I have done sometimes intentionally because it is a lot of work. Okay, you have always to repeat this several times, and you see that the compound 7 was really the best of the class. We started to have photo index up to 140. This is very, very interesting, and you can see that we have done this at different wavelengths, as well as two-photon, as I will show you afterwards. So we have decided to focus on this compound, compound 7, which is once again extremely simple. And I think it is very nice to see that you can do a very simple chemistry and get, as you will see, some interesting biological results. First of all, we wanted to demonstrate that our compound, upon light irradiation, were indeed producing reactive oxidation species. Using ESR spectroscopy and using afterwards DMP or temp, you can see if a stable radical is formed. And if singlet oxygen is produced, it will react with temp, and you will form tempo, which is stable, and you will be able to see it by ESR. The same is true with DMPO, and you will see if these radicals are formed. And what you can clearly see is that only temp gave a stable radical forming tempo, meaning that we are really producing singlet oxygen thanks to the presence of light and our compound. We could once again do confocal microscopy. I hope you can see, but the compound is again going into the cytoplasm. We did not have any other localization, or at least a precise localization in the cells. What we wanted also to do is to see if what we were seeing, by a confocal microscopy was really true. And we use another technique, again, ICP-MS, because of the presence of the ruthenium. We could extract different organelles and see how much of the ruthenium were in each organelle. And indeed, what we were seeing by confocal microscopy was confirmed by ICP-MS. We had really the ruthenium in the cytoplasm and nearly often not in the nucleus, in the lysosome or the mitochondria. Okay, two different techniques showing clearly the same result. What we have done afterwards is to demonstrate that what I have shown you by ESR was indeed true in biology, in living cells. So what we have done here is to have cervical cancer cells. It was simple HeLa cells. We have given the DCFH-DA. We have given our compound. This DCFH-DA can go inside the cells because it's hydrophobic, because of the esterases in the cells. You cleave the two ester, and if you form singlet oxygen, you will form actually a fluorescent dye, which is green. And it is exactly what we have done here. You see, as soon as we shine light, you start to see what is this green fluorescence outside the nucleus, because as I've shown you before, the ruthenium complex is really localized in the cytoplasm. Singlet oxygen is extremely reactive and will directly react where it is. So it's fitting perfectly. We have exactly what we wanted. We have also shown that the compound were very cytotoxic upon light irradiation. So what we have done here, it's again another video. You see that as soon as we shine light, the cells are bursting, and we could demonstrate that it was a mixture of paraptosis and apoptosis. So once again, there is no chance for the cells to survive. We have also demonstrated, and this was a nice result. We have used those little spheroids, once again, normally about 0.5 millimeter in size. And you have to imagine it's like a sphere. We have given the compound to the spheroids. And because we have these two-photon properties of our ruthenium compound, we can really use microscopy to see where the compound is localized in the spheroid, in the sphere. And you really see that when you look through the Z-axis, you have really the phosphorescence all in the whole spheroids. Okay, sometimes you have the anti-cancer drug, which are just going to the crust of the spheroids. This is not the case here. Now, obviously, the most important at the end, can we do it with 1 photon and 2 photon? You see the spheroid growing. We shine light only once. We did once at 500 nanometer. We could have done at higher wavelengths, but this has been done in China and they did not have a laser at higher wavelengths. We shine light and you see that compound 7 really reduce the size of the spheroid. The same happens if you do it at 800 nanometer. And this was very, very nice because once again, 800 nanometer allows you to go really deep into the skin. We have done also in vivo on mice. This was also done in China. You can see that the mice did not suffer during the treatment. They did not lose any weight during the 50 days of the treatment. And what they have done is to give a very nasty types of cancer cells to the mice. And once it was growing, they shine light either with 1 photon, OP, or 2 photon. And they have seen if the tumor was growing or not. And you can see that we can really avoid the growth of the tumor thanks to the 1 or 2 photon irradiation. While if you just have the ruthenium or just the light, the tumor is growing. So you can see here exactly what I have told you, that the tumor is really staying really, really small, or instead of growing very, very nasty if you don't have any treatment. Can we do better? And we were a bit surprised when we have done an extra experiment about the biodistribution of our compound. If we inject the compound into mice and kill the mice and look where the ruthenium is located, and once again, this is a nice part to use metal-based drug, is that now we can kill the mice and take each organs, dissolve it in nitric acid, and determine the amount of ruthenium in each organ. And you can see that actually the compound were mainly in the intestines, the liver, or the kidneys, showing clearly that the mice are trying to excrete our compound, and only a little bit in the tumor. Still, we succeeded to get relatively interesting results, but we wanted really to improve the uptake in the tumor. And this is when Johannes has decided to encapsulate the ruthenium into a FDA types approved polymer. So the idea, you take the polymer, which will wrap around the ruthenium, so that you can inject this into the mice. And because of the enhanced permeability and retention effect of the nanoparticle, you may have a better accumulation in the tumor. He also took a polymer which had a biotin, and he was hoping for a targeting of this biotin through the sodium multivitamin transporter, which is sometimes overexpressed in certain types of cancer. He has done here nanoparticle about 100 nanometer in size. They were extremely stable, this nanoparticle, for several days, no problem. And he did a bit of cell work first this was again in China. And he could really see that once you inject now the nanoparticle instead of the ruthenium in solution by itself, you see that you don't have any more a cytoplasm accumulation, but a typical lysosome accumulation, which is typical for nanoparticles. What he did also is to look if the nanoparticle, which has this biotin, allow for a selectivity towards certain cell line. And he used this A549 cell, which have the receptor, and this HLF, which do not have this receptor. And what he did also is to take cells which are expressing the GFP, the A549, so that they are green. And this one is not expressing this protein. So once he put the nanoparticle, if you start to see the red where the green is, it means that you have exactly what you want. You have a selective accumulation of the ruthenium in those cells which had this receptor. And you could see that there was really a nice selectivity towards those cells, which was exactly what we wanted. Once again, we could use spheroids. Once again, these nanoparticles were really going through the whole mini tumors exactly as we wanted. And finally, we went again for in vivo. And what you can see here, this is really the most important. You have a 8.4 times higher accumulation of the ruthenium compound in the tumor compared to the ruthenium itself. We have injected here, I really want to emphasize this, the same amount of ruthenium once it was in solution, once it was encapsulated into this polymer. We have injected exactly the same amount of the ruthenium, but you can see that we have a better accumulation after 12 hours, notably of our compound in the tumor. We could afterwards irradiate again with 500 or 800 nanometer, and once again avoid the growth of the tumor. You can also see that the mice did not suffer, they did not have any trouble, they did not lose any weight. I really hope during this first part of the talk that I could convince you that such a ruthenium-based photosensitizer have really high promise as a PDT photosensitizer. And I'm sure also that encapsulation can help a lot to increase even further the selectivity, which once again is already given by the medical doctor who shine light really at a selective location in the body. The second part of the talk is completely different. I want to talk about medicinal organometallic chemistry. Sorry, it is one of the projects that I'm working on with Judy, and it is really the use of compounds which contain at least one metal carbon bond. And today I would like really to emphasize the work of Riccardo Rubiani. He was a postdoc in the lab, as well as Jan Lin. She just finished a PhD in Paris a few months ago. We are using such compound to treat different fungal infection, parasitic disease, or even for animal health problem. Now, in terms of fungal infection, I would like really to insist that fungal infection are killing people all over the world, especially the people who are immunocompromised. It's not only small stuff that you can just take a cream and solve the problem. You have really, really serious problem with fungal infection. And you can also see that the drugs which are on the market to treat fungal infection are relatively old. And what is also worrying is that the new compounds which are coming to the market are quite often a derivation of on a theme. Okay, it's a known compound which has been derivatized, and you really need new classes of compound. You see also that people are working on this topic, but it is not growing like, for example, it is growing for anti-cancer. What is very worrying is that the number of resistant strains which are discovered every year. We have decided to work on this topic because two actually French scientists discovered separately a very simple thing. If you take an organic drug like tamoxifen, which is used, it is a frontline chemotherapeutic treatment to treat women with breast cancer. If you replace one of the phenyl by this ferrocin, and I will insist that ferrocin, you can buy it, it is absolutely non-toxic. You can do exactly the same chemistry that you will do with benzene. Okay, you can do also a disalder or whatsoever. If you replace this phenyl by this ferrocin, you suddenly have a compound here which is active on cells where tamoxifen is not active. And the same has been found with chloroquine. which is a known anti-malarial drug. The only trouble of chloroquine, you have a lot of resistance. Now, the people in Lille in France have replaced have changed this lateral chain by addition of a ferrocene moiety. And you see that ferroquine, which has been bought by Sanofi, which is currently in clinical trial, this compound is really active on chloroquine-resistant strains. And just by changing a little things in the structure, you get something very interesting. And this has been really linked thanks to the production of reactive oxidation species, for example, through Fenton reaction. So what we wanted to do is exactly the same, but with a fungal agent, antifungal agent. You see that clotrimazole is very well known, but we were extremely interested in fluconazole. And this is why we have prepared those type of fluconazole ferrocene derivative. We have replaced one of the triazole by ferrocene. Now I want really to rationalize this choice. Okay, we did not choose this part only by chance, but if you look at a model which has been made by Chai, you see here a P450, that's a P450 with iron in the middle. You see that fluconazole is binding covalently the iron of this P450. You see afterwards that the two fluorine are really involved in the interaction with the enzyme, while this triazole here seems to dangling around and not to have a precise role, potentially on the biodistribution, but not on the binding. And this is really why we have decided to replace this triazole with a ferrocene. So here, you have really 14 alpha demethylase. Now we have done all the compound. I'm not talking about the synthesis because that's not the point of this talk, but we have sent this to Italy, to Tobias Weil, and we were extremely happy to see that our compound, especially the number 2, was extremely active on so many different strains, where fluconazole was not at all active, where there were so many problems with resistance. We have even cases where our compound was 800 times more active. And Tobias was directly very excited and told us, oh, this is very rare, I've never seen this. Now I'm just worried that your compound is just a killer, it's just killing everything. And we have directly tested this on different healthy cell line, MSc RP1, and you see that our compound is as a non-toxic or relatively non-toxic, which is comparable to fluconazole. Okay, we did not just made a serial killer. There is just really something that we don't understand yet, why this compound is so active on these strains. We had a look at the enzyme inhibition properties of our compound, and we were extremely surprised to see that our compound is inhibiting P450s, different P450s, really much better than fluconazole. And this was really a surprise because, as you can see in our compound, you have really the triazole which can bind to the iron of the P450, but it doesn't explain why you have such an increase in activity. We just could see also that our compound, as expected, were more lipophilic. Okay, the presence of the ferrocene moiety is really increasing the lipophilicity of our compound. We have done with Tobias Weil and Marco Moretto chemogenomic studies to see if the target of our new ferrocene compound were indeed the same as fluconazole. And we could really see that this ERG11, that's the one encoded the lanosterol 14 alpha demethylase, which is a target of fungal agent, like fluconazole, are indeed the same for a compound. And this was really, really interesting, showing clearly that we are targeting at least the same target than fluconazole. Now, what we were hoping is that actually our compound are producing reactive oxidation species because of the presence of the ferrocene. And unfortunately, this was not the case. We see that as many reactive oxidation species are produced with our compounds and with fluconazole, not explaining actually why we have this nice activity. I have presented this work a few months before, and my colleague John Spacer directly told me to get in touch with Anthony Moore at Sussex in England, because possibly the target of our compound will be the alternative oxidase, AOX, which is really typical for antifungal agent. And we have looked at this, and unfortunately, this was not the case, saying that it is something else which explain actually the high activity of this compound. We have done TEM studies, and we can really see that fluconazole and 2A have a different mode of action. We can really see a loss in wall integrity. This is Anders Kerr at the University of Zurich who has done this for us. We have done also in vivo, and this is really Luigina in Italy who has done all the work. I don't want to go too much into detail, but what we can really see with this work is that our compounds are really able to reduce fungal growth. In this case, we have used a strain of which was not resistant to fluconazole. So we have used fluconazole as a control. So we have similar results than fluconazole, which is really, really good. But what was very, very interesting is that our compound, contrary to fluconazole, really promote a strong pro-inflammatory cytokine response, okay, which was really, really nice. We have a decrease in inflammation, and this was really something that was of high interest for Luigina, and we are currently trying to do new in vivo experiment where we will try to use this time a fluconazole resistant strain and see if our compounds are still very, very active. And this is when we have received the grant with Judy to see if such compound could be active against other parasites. Currently, Kilian, which is paid through this grant, is working really hard to make new compound and to try to understand how things are going. Harley, an Australian postdoc, is really helping him. And I really thank Kevin Carey, who is a CNRS researcher, which is also working with us on this exciting project. Finally, I would like really to emphasize that the use of organometallic compound should not be only for catalysis, but that there is a lot to learn, for example, in terms of medicinal chemistry. I would like really to thank all the students in Paris. I was talking with Gina and Judy before, but we have really an international group. We are really blessed to have people from all over the world, and they're really doing a fantastic job. I would like also to thank all the students who have worked before in the group. They have done really so much for the group, and they are all doing really, really well. They have all found some work in industry or in academia. And I also would like to thank the people that we are collaborating with. You know, there is Judy here, and finally the people who are giving us money. And I would like really to highlight the PSL Qlife grant, which allows me to be here with you today. Thanks a lot.
Wow, excellent talk. Thank you so much. It's really a lot of work that you presented here.
Thanks a lot.
So for the listeners, if you could just please put your questions into the Q&A box, and then we will read them off. So go ahead and please do that. We have maybe about 10 minutes. And perhaps I could just sort of lead off with a question about some of these compounds for the parasites. And I know we almost got into this with the Trypanosoma cruzi that causes Chagas disease, and some of your compounds that were, I guess, analogs of the posaconazole.
Yes, so yeah, as I was telling you, it seems that some of the compounds are really working on T. cruzi, which is not a big surprise because T. cruzi has the same enzymes than, for example, the fungi have. But it's always nice, you know, to have two different models and to see that you get still nice results. I have to see with T. cruzi for the moment, it was not on in vivo. Okay, this has to be demonstrated also in vivo, and this is what we want to do in the future. But yeah, there is definitely something more to dig in, I would say, in this project, and I think it's only the start of a project which can last for several years. I hope so.
Yes, especially since the Trypanosoma cruzi, you know, it's related to all these other trypanosomes that cause devastating diseases like the leishmania and as well as the African trip. So it will be really interesting to see what comes out of that as well.
Yeah, so it's what I was discussing with Adam before, but he was telling me there was one of these compounds which was in clinical trials against T-Cruzy. I will have really to look which one exactly it is and what it is at the moment, if it made it or it did not make it to clinical trial. That's a good stuff to come here. You are learning a lot of info.
Yes, absolutely. And then if you could just talk a little bit about these azoles, these organometallic compounds that we're using for our worm project as well. So if you can just talk briefly about that.
Yes, hang on, I will just try to go back if I can. Hang on, I think I will have just to stop and start to share again. Yeah, here, hang on. Oof. Oh, bizarre, sorry. I thought, why it's not. And I don't know why it's coming. Here, sorry. Yeah, so that's this woody ferrocene compound here, where you can really see that we have just replaced the triazole with a ferrocenyl moiety. Now you can see that there is much more that we can do. We have started to put other types of compound here. Also, what is very, very interesting, that's why I have said that we have Harley's Australian postdoc. We are currently trying to understand if metabolism can explain, the results that we are getting. We are currently trying to see if the compound are stable in human plasma, if they are transformed into microsomes. We have both microsomes, and we are currently it's really this week that we are currently doing this to see if the compound are transformed and if the different activity we get, because it was very interesting. I think we have sent you about 15 different compounds, and you have really activities for certain, and other had absolutely no activity. Also, the design was exactly the same, and there was maybe just a little maybe methyl which was different compared to the other one, and you have a total loss of activity. And we hope to see that one of the compound is transformed to something while the other one not, and this will explain actually the activity. This is really what we are trying to do now, and we hope to see some trend very, very, very soon. And yeah, as we are discussing with Judy, but we hope to have some in vivo results, hopefully in the next couple of months here in America. And it will be interesting to compare those results with the metabolic studies that we are currently doing now. Does it reply to your question, or you had something else in mind?
No, that's perfect. I was just curious too. How do you select which particular metals? Like here.
Yes. So technically, when we are working with such disease like fungal infection, we want really to use a metal which is bioavailable, like iron. If I'm telling you that I will give you some iron, you won't be scared. People won't be scared. We want to use, for example, something like ruthenium or something like of palladium, I think, because it is normally a treatment that you will have to take for several days, weeks, or months, an antifungal treatment. And we talk about anti-cancer, normally it's something that you will get hopefully never in your life, or if it is the case, only once, okay? So there won't be a problem of accumulation or problem of toxicity over the long term. That's why we are using, ferrocene, which is already, in clinical trials. So there is no problem of convincing a pharmaceutical company that it's dangerous. Okay, we are really considering all those things when we are designing a compound. And the most important reason why we are using ferrocene, it's because you have this redox activity. As soon as you change the iron with another metal, it seems that the redox activity is fully different, and you cannot have these phantom types of reaction to produce reactive oxidation species. So if you have H2O2, so water peroxide, hydrogen peroxide, sorry, I was looking in German, hydrogen peroxide, you can form OH point. And this OH point is extremely toxic to the cells. And it has been really shown this with ferroquins that OH point is formed in the parasite. So you have in the parasite hydrogen peroxide, and because of the presence is iron 2, you can produce OH point. And we were really hoping to get a similar effect in the fungi. It seems it is not the case, okay? This has to be confirmed, but I have really shown you those results where the reactive oxidation species studies, we see a bit at the beginning if you look something a bit higher for 1A and 2A, but afterwards, after 18 hours, you have more reactive oxidation species fluconazole compared to our compound. So we are not really convinced that this is a reason for the overcoming the resistance, okay? Because we are definitely overcoming resistance we are compound, but we don't know why. And once again, I'm always very honest when I'm giving this talk. I have given this talk over the last few months, and I'm always saying, if you have a good idea, if you believe that you think that there is maybe something that we should do, please contact me. We are more than happy to send you the compound and to explain this point, because for the moment we have to say we are a bit lost in the sense of we are happy because we are overcoming resistance, but we cannot rationalize it.
Okay. We'll have another question for you. You know, in terms of you know, I think in terms of giving drugs for many of these neglected tropical diseases where they occur in low and middle income countries. How expensive is it to make these, to synthesize a lot of these metal compounds?
This is also one of the reasons, because we have worked also a lot of those neglected diseases like schistosomiasis. Technically, the ferrocene is extremely cheap. So this will never be an issue. To make the compound, if fluconazole can be given, we can give this compound. It is extremely simple. And I really insist that the synthesis of this compound, I think it was a few step synthesis, and this is really key when you are making a drug, is how long it takes to make the compound, and you know how many synthetic steps. And this is really the same than fluconazole. You are just replacing one of the triazole with this compound. So this will never be an issue. We could go to any foundation and rationalize this. This is really one of the good points of this ferrocene. Very easy to make, non-toxicity. And yeah, cheap.
Great. I see Adam has his hand raised. Adam, would you like to just pose your question?
Yeah, can you hear me?
Yes, thank you.
Great talk, Gilles. So with this set of compounds, I'm wondering if you've made sort of control compounds where you replace the triazole with maybe a pyrazole, so you get rid of the nitrogen that can ligate to the CYP. And just to kind of help you identify if there are additional targets or other mechanisms of action, because if you remove that nitrogen that your laser pointer's on, that will no longer be a CYP inhibitor, right?
Yeah, that's a good point. That's something that we could easily do. My only trouble by doing this, you know, if you put a methyl, for example, here, it will become positively charged, and I hope that it not change, you know, all the cellular uptake or on something like this. But maybe we could just do it, you know, on the inhibition study to see if we completely quench the inhibition. But yes, that's something that we could do. Thanks a lot, Adam. That's very easy. Should we also methylate this one? Do you think both, to be sure?
I wasn't thinking about N-methylation, but rather just replacing that nitrogen, the distal one, with a CH, just a pyrazole ring, right?
Okay, just even easier. Yeah, that will be a good point. Oh, I will ask Kilian to do this to see if yeah, that will be a very easy. And if we still have an inhibition and still have an activity, it clearly means that there is something else. It will not tell us what, but it will clearly mean that there is really an additional mode of action. Thanks a lot, Adam. Yes, that's very nice.
Yeah, just to share one anecdote. When we were working on some protease inhibitors quite a while back, of you know, cathepsin-like cysteine protease inhibitors of a parasite, And in one of the distal ring systems, we just added a pyridyl, a 4-pyridyl ring. And with that one change, we started picking up activity against CYP51.
Yeah.
It wasn't at all designed to be it. So those sort of single atom changes can have huge effects, obviously, on these enzymes where you're ligating a heme cofactor.
Yeah.
Yeah.
No, that's a good point. Yes, thanks a lot.
Great question. I see it's after 3, so I just want to thank the viewers, the audience, and thank you for coming to San Francisco and giving this great talk. So thank you again. Thank you, QBI.
Thanks a lot.
See you.
See you.
Okay, bye.
Bye.
Au revoir.
Right.